Okay, so I'll be walking you through a number of simulations that I have been doing on organic matter interacting with mineral surfaces and with ions in water. Um, and Before I start all this, I will stress the importance of studying soil organic matter and soil in general. Soil is the largest terrestrial pool of global carbon, um, and uh, and it is also important for our food and ecological security. And and a molecular level structure and dynamics of soil organic matter is not well understood, and so bringing a classical molecular dynamics or quantum chemistry level detail to this problem provides better insight into global carbon cycling modeling, especially looking at multi-scale modeling can bring some insight into that and insight into soil nutrient and micronutrient cycling. So this particular example is about, well, the research was originally part of an airborne soil organic particle research campaign at EMSL. And so soil can find itself in the air and from irrigation and rainfall. And this is in collaboration with Alex Laskin, who collaborated with me with this work. So to get an idea about what molecules to compose a soil organic matter model out of, there are some studies out there that give you a rough idea about what biopolymers you have or biomolecules you have and other organics in the soil. But this composition of soil is going to depend on vegetation, microbial communities, geology, climate. So we could only go with a rough estimate of the composition of soil, and we also needed to take a reductionist approach where we couldn't put everything into the model. We wanted a zeroth order model where we just took 4 components, carbohydrates, lignin, protein, and lipids, and formed an organic model. So from these representative, 4 representative compound groups, we chose a very common motif of lignin. This is a fragment of lignin polymer. Trehalose sugar, which is a common osmolite sugar that is produced by microbes. Palmitate fatty acid lipid, which is also commonly produced by microbes, and pentaglycine peptide, a very simple representative for the protein amino acid or peptide group of compounds. Now, there are other compounds that we could also incorporate into this model at a later date, including tannins and amino sugars and others. But we started with a very simple model. And the relative amounts of these 4 compounds are in line with the amounts of carbon, nitrogen, and oxygen found in NWChem's analysis of airborne soil organic particles that my collaborator Alex Laskin took. And so that's the organic matter composition for the model that we are putting together. And so we wanted to interface it with a number of substrates. And we chose cellulose 100 surface and 010 surface. The cellulose 100 surface is somewhat hydrophobic, and the cellulose 010 is quite hydrophilic. And we use this to represent the nanostructure of intact plant matter that hasn't broken down yet. And we also interface it with, we chose clay mineral montmorillonite, either sodium or calcium exchanged, to represent our mineral model. And so I'm not going to go into a lot of detail with how we did the classical molecular dynamics, but I'll say that we use the amber compatible force field for all of the components in the system. We use NWChem to get atom charges to assign to the lignin atoms. And we use LAMPS, molecular dynamics code, to run these calculations. We could have used other codes, but LAMPS is especially flexible with dealing with these heterogeneous types of systems. So here I'm showing snapshots of the 4 different surfaces that we simulated, and I've color coded them according to what kind of compound you're looking at here. So the red is the lignin fraction, the blue is the trehalose fraction, the green is the peptide fraction, and the yellow is the lipid fraction. And we found that these compounds readily aggregate and adhere to these surfaces. And if we take a closer look at what is going on with these organic compounds on these surfaces, we look at the density as a function of distance from these surfaces. We see that as expected, the cellulose 100 surface shows its hydrophobicity, with the lignin fraction having a high density at the surface of that system. And interestingly, the hydrophilic protein also shows us a fairly high density there too. There is the potential for hydrogen bondings that could take place with the surface by breaking some of the hydrogen bonds in the surface. And if you look at the 010 surface, which is more hydrophilic, you'll see that the lignin, the hydrophobic lignin, is its density is pushed out a little farther from the surface compared to the cellulose 100 surface. And with respect to the mineral surfaces, with the sodium montmorillonite, we see that the hydrophobic lignin is not really interacting with the montmorillonite surface, and that's expected since there's a lot of water pooling up at the surface, along with the charge of the system being negative, and and the sodium ions also aggregating around this area too, which create a quite hydrophilic wall for that lignin to interact with. And we also find that in general, the soil organic matter broke in two, and part of it was on the upper surface and part of it was on the lower surface. And with respect to the calcium montmorillonite, we see that the lignin is interacting a little bit more with the montmorillonite surface, and we believe that that this is a little more complex because you can have calcium bridging, and so we're looking at this a little further. We find that the organic matter aggregate is more intact when you compare it with the montmorillonite sodium surface. So we also looked at the water density of these systems and found that there is water that has been entrained inside of those aggregates, and at various degrees. So, for instance, we looking at the montmorillonite system, we see that there's this trough here where there is not a lot of water inside the core of this, because the core is actually has quite a bit of lignin and the lipid inside of it, whereas it seems to be a little bit more uniform in density when you go and look at the density for the calcium montmorillonite associated organic matter.
Amity, this is your 2-minute timer.
Okay, thank you. So I'm just going to run some of the dynamics of these systems to show you a little bit more of what's going on. We find that the lipid tends to stay within the aggregate, within the lignin fraction, and we also find that especially with the peptide, that it tends to solvate more readily during simulations. And we wanted to look at that a little closer, the solvation of these systems. So this is just showing the frequency of solvating each one of these organic components. And I'll just go through the peptide one here and look at the sodium montmorillonite. And what you see is that it's quite frequent that you have the peptide solvating. And if you compare that with the calcium montmorillonite, you see that the peptide still solvates quite readily, but it's less so with the calcium montmorillonite compared with the sodium montmorillonite. And we believe that is the calcium bridging that is occurring in that system. So this is showing just in the shaded areas the entrainment of sodium ions and calcium ions in the system. They don't seem to diffuse that readily out into the water during the simulations. And we want to look at these, the environment, the local environment of each of these ions a little bit closer. So we are taking that smaller cluster of water and organic matter out of the system to look at with NWChem's TDDFT module, the XANES of these particular cluster systems. And this is just an example of a few of many different clusters that we can grab from one of these systems and produce, like the sodium XANES and the calcium XANES from these clusters. So I just like to conclude that we saw that we got aggregation of these organic systems that are quite heterogeneous mosaics of hydrophilic and hydrophobic domains that have an interplay of ionic, hydrogen bonding, polar and non-polar interactions going on, and that the organic matter readily entrains sodium and calcium, and on chemically similar surfaces like the cellulose, the organic matter readily adheres, and we find that there's less adherence of the organic matter on the sodium molynides system compared to the calcium system. And we're exploring the cationic bonding of the calcium system a little bit more. And I'd like to acknowledge the research campaign for airborne soil organic particles at EMSL, and my collaborators, Sharon Bone. We're exploring more calcium coordination systems through this molecular dynamics approach, and of course Alex Laskin, who I have been doing this airborne soil organic particle research with, and my collaborators Mira Govin, Ravi, Odetta Person, and the NWChem team. And I just want to note that we are looking at other surfaces with my other collaborators like magnetite, calcite, and illite. And I'd like to acknowledge the computing time at EMSL for this work. And thank you for your attention.